Good morning, everyone. Today we'll discuss about thyroid hormones and thyroid enzymes. So as you can see from here, thyroid gland is situated the neck region is present just below the phone line, just above the liver. Now we'll discuss about types of the thyroid hormones. So basically there are three types of the thyroid hormones. First is T4, that is thyroid. Next comes T3, that is triiodothyronine. Then calcitonin. So when we talk about T3 and T4, these are the protein, but apart from this, T3 is that of T4. It is produced in less amount as compared with the T4. Basically, function of T3 and T4 in the body is associated with the growth, development, as well as the energy metabolism. Now we talk about calcitonin. Calcitonin is the third hormone which is produced from the thyroid gland. Calcitonin is being produced from the interfollicular cells. When we talk about the interfollicular cells, in the thyroid gland, there are thyroid follicles as well as interfollicular cells. Thyroid follicle is the main part of the thyroid gland. Interfollicular cells are the regions just in the environment of the thyroid follicles. So in the thyroid follicles, calcitonin is being produced. Calcitonin is associated with calcium metabolism and it is used very on regions just to politics. Talking about the historical significance of the thyroid gland. So let's discuss it. Thyroid gland was been recognized in the 19th century and raised at best of scientists were associated with most of the thyroid gland. Living in large these connected nephron with insulin. Now, insulin was one of these conditions which is due to the thyroxine. T4 was the first form to synthesize the laboratory. Then its chemical structure identified by Kenny in 1950 and the confirmation was done in 1926. So these are the historical perspectives of how thyroid gland was produced and which were the scientists which were associated with the discovery of the thyroid hormone as well as thyroid gland. Talking about the synthesis, storage, and secretion of the thyroid hormone. So when we talk about the synthesis, storage, as well as secretion, before that we need to know the constitution as well as structure of the thyroid. Now when we talk about the thyroid gland, there are different cells as well as the regions which we need. So the main region, the most important region is the thyroid follicle, as well as it is also known as the actinium. So within the thyroid follicles, thyroid globulin, which is a glycoprotein, is very accumulated. This thyroid follicle is surrounded by the epithelial layers. Apart from that, the main region which is been shown here is the interfollicular space to which the calcitonin is talking about the synthesis of the thyroid hormone. First is the step of iodination. Now, when we talk about the synthesis as well as the storage of the thyroid gland or the thyroid hormone in the thyroid gland, first we need to know what are the chemical structure of the thyroid hormone. So it starts with the amino acid, tyrosine. Now, tyrosine amino acid, which is being conjugated in thyroglobulin, which is a glycoprotein. So in this, you see the chemical structure of the tyrosine with different chemical or subgroup of amino. Now, when the iodination of tyrosine happens, so in the amino acid tyrosine, you can see the thyroglobulin has been associated with the amino acid tyrosine. Now, when the iodination of the tyrosine happens, the iodine is concentrated in the place of the hydrogen in the parabolic. When this conjugation happens, it forms monoiodine tyrosine. But when the two iodine resins are being attacked with the structure of this tyrosine containing the hydrogen, tyrosine will produce. The significance of the monoiodine as well as tyrosine will be discussed from the sense. Talking about the synthesis of thyroid hormones. Now, when we talk about the thyroid hormones, these individual structure associated with the two iodine residues are not sufficient. So, in this, the coupling of the thyroid specifically monoiodine as well as thyroid takes place. Now, when we talk about the coupling, basically, when the two thyroid residues conjugate with each other, they form tetraiodine because there are four iodine residues which are attached at this structure. And we talk about the conjugation of the one monoiodine that is one diiodine takes place from triiodine because the moon will consist of the one iodine residue, while the diamond consists of two iodine residues. So it will form that triiodine. specifically T4 in other studies. When we talk about the mechanism of the synthesis of thyroid hormones, we need to see which are the distinctions, the structure of the thyroid gland. There is the plasma follicular cells of the human of the follicles. So in the plasma, when the iodide is minus the concentrate, the concentration of the iodide takes place in the process where it's in something you said today. It's concentrated iodine will maintain the follicle cells. Now, since there is a co-transporter known as NIH, which is a sodium iodine film motor, comes into the method. This iodine gets entrapped into the follicular cells. After entering into the follicular cells, gets passed with the lumen of the follicular cells. Lumen of the follicular cells will consist of myoglobin as well as there will be a row of iodines which will iodines iron I minus because of iodine. Right? So after this, iodine entering into the follicular nucleus gets converted to the iodine. Now this iodine gets associated with thyroid. That's the same thing. Right? So you can see here, TG, which is a thyroglobulin, is associated with the dialysis. Here, this dialysis consists of the iodine density, which is treated iodized by the thyroid and H2O. Along with that, monoiodinosin and diiodinosin are also there. Now, when this conjugation takes place, this complex enters again into the follicular cells. After entering into the follicular cells, this complex simply lies by the lysosomal enzymes. Now, lysosomal enzymes, what they do is they cut off the amide as well as the individual calcium residues which are not written down. So here, before entering into the follicular cells, amide and DID, when coupled with each other, either they will form T3 or either they will form T4. What there will be the rest of the calcium residues which are not with terms of these residues will be separated from this complex. T4 and T3, which are not coupled nearby, the amide and DID will be released. This T4 and T3 will enter into the system itself. While the non-coupled calcium residues as well as the different residues which are on the side are being gained into the cycle. So this is how T4 and T3 are present. Now, talking about the regulation of the thyroid function. Now, if we talk about the thyroid function, there is a role of the hypothalamus as well as the pituitary. So, if you see towards this diagram, you can see there are different stimulants which take place in the release of the thyroid. So, first there comes the role of the core trauma upset. So, these are the stimulators in fact, which leads to the further signaling towards hypothalamus. Hypothalamus, what will do? Either it will release somatostatin, which is an inhibitory hormone, which all the hormones which are produced by the pituitary gland, or release the release of the PL. Releasing hormone. Now, talking about the releasing hormone, function of the releasing hormone stimulate the pituitary gland, specifically here the anterior pituitary. So you can see from the indications, somatostatin shows a negative regulation, while the TRO, thyroid releasing hormone, it triggers the release of the thyroid hormone from the anterior pituitary. Right. So after the release of the thyroid hormone stimulates the thyroid hormone production, as indicated by the positive sign. So why thyroid gland has been stimulated by the thyroid hormone releases T4 and T3. Right. So C towards this indication T4 and T3. Right. Here, conversion of the T4, the T3 will take place in the system itself because T4 is produced with a greater amount as compared to T3. But as comparison from T3 and T4 to T3 is more potent than T4. That is why conversion of the T4 towards T3 will take place ultimately. There are the different kinds of drugs as well as the components which affect this genetic marker. The first comes to the protothelium. Protothelium is one of the drugs which affects the release of the thyroid hormones indirectly. Because it triggers the TRH activity causes the increase in the phosphate mechanism, the leading to increase in production to greater amount. Next, talking about the iodine in excess. Now, as we saw from the mechanism previously, iodine is essential for the synthesis of the nucleic gold from the gold crystal cycle. We get to know that. But when the iodine is produced in excess amount, it causes a negative loop, which causes the inhibition of the nucleic gold level. Here you can see by the negative excess of the iodine, it stops the thyroid hormone for the production of nucleic. And next is thyroid. Thyroid are part of the blood, fighting with the inhibits the T3 zone by directly acting on the thyroid gland. Next is the 131 isotope of the iodine, which is it shows the mechanism by the destructive way. It ultimately causes the destruction of the thyroid follicular cells, which ultimately leads to decrease in the hemoglobin of the component. Next is the thyroid hormone T comes. Also, this is the thyroid hormone coming associated with metabolism first, growth, whereas level. When talking about the metabolism, T3 T1 are basically immediately associated with the metabolism in terms of the DNA. That is the basic metabolic rate. So what they do is increases the oxygen consumption as well as the heat production. So oxygen consumption will increase the ATP generation, which will be once the ATP generation is going more, its energy side, that is the heat production, will be increased. Basically, these processes are been influenced with conditions such that parts, member, and muscles, but now have not been regulated. The brain, connect, as well as explained. Now we'll discuss about the mechanism of action of the thyroid hormone. Up till now, we saw how thyroid hormones are produced, how it's affecting different functions in our body. You see, they are able to see the cellular levels, such as the nuclear, how does it affect the stomach? Now in the bloodstream, T3 is one of the specific one. From T3 is more potent, it is the only portion of the thyroid hormones which is very different. However, the conversion of the T4 to T3 will give with the help of the thyroid iodine. So once it gets converted to the T3, entered into the cytosol. After entering into the cytosol, enters the nucleus. Within the nucleus, there are nuclear receptors, such as the insulin. Here the TR represents calcium receptors of the receptor. TR represents the thyroid response allele. It's the portion of the DNA which is responsible for the transcription of the proteins which are necessary for the functioning of the thyroid. Here, RX retinal receptor is fusing for the attachment towards the TRL. What happens? The T3 enters the nucleus, they expand to the specific portion of the thyroid receptors. Here, also apart from that, coactivator is the coactivator is the co-repressor of the portion of the proteins which are associated with the thyroid response, LED, as well as the TLs. So what occurs in normal condition, co-repressor is implemented. The function of the co-repressor is to suppress the activity which is shown by the DNA as well as the TR, that is thyroid resonance. What occurs is the T3 gets bound to the response element or the portion of the thyroid resonance. The co-repressor is in place with the co-activator. So once the co-activator takes place, the response elements are triggered and the transcription of the mRNA takes place. Further, for the mRNA, the proteins are on the side. This proteins will get activity as well as stomach. Next is thyroid disorders. There are different thyroid disorders that are associated with the levels of thyroid hormone. The first is hypothyroidism. Hypothyroidism is a condition in which there is an increase in the thyroid levels just to T3 to So hypothyroidism when disease news and animals caused by the insufficient production of thyroid hormone by the thyroid gland. There will be increased production either the restriction of the thyroid follicle or inhibition of the environment of the thyroid as well as other elements which will be shown in the further slide. So what are the clinical characteristics of the feature shown in the hypothyroidism? The first one will be fatigue, that is increase in the energy cell. Next is depression, modest AD to be shift in the cholesterol metabolism. Next is cold because increase in the thyroid hormones to lead to the imbalance normal energy metabolism as well as thermal stress body. Excessive sleepiness, close neck, constipation, dry skin, muscle cramp, increase in the cholesterol levels correlates with the modest weight gain, increase in the concentration, swelling of legs. These are all the basic characteristics that are shown by hypothyroidism. So when we see towards the diagram, especially in the face of the person, we can see the high pressure person is blended. There is a deposition of the uneven deposition or the struggle of fats all over the face. So there is a sense of depression of the face as well as it moves and windows and Next is hyperthyroidism. So as contrast to the hypothyroidism, hyperthyroidism state in which there is an increased production of the T3 against T4 to manage immunism. So the clinical characteristics of this condition is to accompany one appetite, the weight loss, palpitation, heat intolerance, nervousness, insomnia, breathlessness, increase in the bowel movements, line or the absence of menstrual periods for the female system. Talking about the males, there has been related to retraction, gland, extra hormones, the bulging out of the eye, and the chemosis awareness. Of challenge is the awareness towards the xylem, the strong prayer. If this phase in pregnancy or symptoms, as well as the clinical representation of the genetic hormone. So there was retraction. Here, you can see there is a chance of absorb hormones. There is a bulging out. So the cell eyelids are made reconstructed. Here you can see that the hairs which are present there are totally different as compared to what hypothyroidism can be. So this was all about thyroid hormones, its synthesis and how they function. I hope you have a clear idea what we have discussed. Thank you.